Good morning, everyone. Can someone hear me from their side? Can someone hear me from their side? Maybe you can raise up your hand or send a message, a chat, so that I can start sharing on my screen. Okay, I've seen Boniface. Boniface, I'll ask you to unmute and speak. Can you hear me loud and clear?
Yes, I can.
Okay, fine. Now, because we've already lost 3 minutes, I think we will just begin. So, with consultations from the department, we did discuss with Mr. Hassan, and I was told to discuss with you this topic called growth and development. So with me here are some questions that I want us to go through. And I really wish this is going to be a very interactive session where we look at these questions and try as much as possible with precision, answer them, and also get the content on those questions that we are going to discuss. So good morning, everyone, and welcome to this class now. So I'll present my screen straight away. So I also hope that I believe that all of you can see my screen, and these are the questions that we need to discuss in this class today. So if anyone is having responses, we can be looking at your responses from the chat side or also from those that which require explanations. We can get them from individual raising up their hands and also speaking out their minds on those said questions. So we begin from question number 1, and this is a question paper that only talks about growth and development. So in as much as we are going to answer our questions, we are going to at least try and confine our contents and responses to growth and development, since at least first trying to identify where a question is coming from, that is from which topic, it makes it easier for us again to try and reason out our ideas in relation to that. So the first question is, and I hope all of us can see my screen. The first question reads that name 2 types of curves obtained during measurement of growth in living things. I can see Tim Collins has raised up his hand. So I can see so many people. So let us get responses. Yes, Tim Collins, you can unmute and answer that.
Sigmoid curve and intermittent curve.
That is very good. So we have those 2 types of curve. So growth can be measured while development cannot be measured. So when we measure growth, we come with a consistent growth curve called a sigmoid curve, and an inconsistent type of growth curve that is called as an intermittent curve. So those curves that you mentioned, Tim, in which phylum do we find such growth curves? Still on you, Tim? Yes, I can see someone else has raised up their hand. This is admission to at 26917. Please, you can unmute.
Philomanthropoda.
To which curve?
Which?
Which curve are you referring to? We have two curves, intermittent and sigmoid.
Intermittent.
So for intermittent curve, we have phylum Arthropoda. What about Arthropoda, not Arthropoda? What about sigmoid curve? You have gone MIA. What about a sigmoid curve? Where do we find sigmoid curve majorly? Of course, in classification, we do only two types of phyla. Yes, I can see Larry has raised up his hand. Larry, please unmute and give us the response.
Phylum Chordata.
So we have phylum Chordata, and I do hope in your responses, you start writing, you follow the rules of writing those names the way they are supposed to. So from the 7 taxonomic units that we have, it's only the species name which we follow the rules of binomial nomenclature. But all other tags are the 6 that are remaining, all the way from kingdom to genus. When we write those names, we start them with capital letters. Question number 2. Name the chemical substance in plants that, 1, promote ripening of fruits, B, flowering in plants, and C, leaf fall in plants. So which chemical substances are these? Yes, I can see Larry has raised up his hand. Larry, you can unmute and speak.
So A is ethylene.
So A is ethylene. And what about B?
B is florigen. And
B is
C is ascorbic acid.
Ascorbic acid, no, that you failed. For the first and the second one, those are very right. We have ethylene. And before maybe I proceed or I talk about all these, let someone at least answer the last one. So we have Enos. Enos, please unmute yourself and speak.
Abscisic.
Yes, it's called as abscisic acid. I think that is what Larry was trying to confuse with ascorbic acid. Ascorbic acid, that is in, when we talk about the various types of food tests that we have, ascorbic acid is also known as vitamin C. So these are plant hormones that promote or that help in various functioning of the plant. And for ripening of fruits, this one, I think it's the most easiest one, because most mama boggers, if they want to ripen for us the fruits that we buy in the market, you'll see some good fruits so shining on the outside, very, very good oranges. But from the inside, when you go and cut them so that you can at least water your throat, they become very, very unbearable. So that would be ethylene. Then flowering plants, of course, that would be florigen, and for leaf fall, that is abscisic acid. Question number 3. State the possible application of the following plant hormones in agriculture. Roman 1 is auxins and Roman 2 is gibberellins. So for these hormones, they have several functions, and we can give as many as possible. So let's start with auxins. What are the main functions of auxins? Yes, I can see someone here called Aldi TKN. Yes, Aldi?
Okay, so auxins are the faster maturity of flowers, which leads to earlier flower formation in plants. They also are used in decapitating shoot tips to allow sprouting of lateral buds.
Okay, your first point, we can go for it, but the second one, I think it has an issue. If you talk about decapitating of buds, that is the removal of auxins. So instead of putting your response in that way, you could have said it promotes apical dominance. Or in other words, you can talk about it inhibits lateral bud development. That is a very good one. Let us give other people also a chance to speak. I can see here Tim Collins. Yes, Tim.
Auxins promote the development or the formation of woody tissue.
Of woody.
Yes, woody tissue.
Which woody tissue? Maybe an example of an woody tissue.
Oh, lateral bud.
Lateral bud. That is not so much clear. Let us give other people also chance to speak. We have Skylar White. Please unmute. Sorry for that. Unmute. Skylar White.
They induce parthenocopy.
Yes, fruit formation without fertilization. That is parthenocopy. That is very good and straight to the point. Yes, Boniface, you can unmute. There are so many.
Auxins promote development of adventurous roots.
Yes,
Adventitious roots.
Yes, adventitious roots, not adventurous roots. That is okay. Any other? We have so many. So auxins, guys, let us remember that auxins also. Yes, Boniface. Sorry for interruption.
They also keep flowers fresh.
Yes, they keep flowers fresh. Any other? Okay. We have auxins. Yes, Denzel also has to say something, but we won't be taking too much time in one question so that at least we also tackle the others. Yes, Denzel, you can unmute.
They stimulate cell division and elongation, leading to primary growth.
That is very okay. And thank you for that. So I won't be accepting any more responses. I know we can be having so many functions of auxins, but amongst them, number one, we talk about promotion of cell division and elongation. We also can talk about it promotes parthenocopy, and that is fruit formation without fertilization. We also talk about promoting formation of abscission layer, and that also helps to bring about leaf fall. Then also it causes apical dominance, and by that it inhibits lateral bud development. Also, we can talk about promoting the growth of adventitious roots, like said by someone here. And also, when we combine indole acetic acid, and that is an example of auxin, and cytokinins, both of them, they induce formation of callus tissue that helps to heal wounds in plants. Then the other hormone here, of course, is gibberellins. And for gibberellins, I think I see someone raising up their hand here. Maybe they have a question to ask. Yes, Enos. Enos, you can unmute. I've seen your hand raised for long.
Wanting to answer the second.
Oh, the second one, no problem. So what are the functions of gibberellins?
Reduce leaf fall by formation of the abscission layer.
Any other?
They promote formation of side branches or lateral buds. From lateral buds,
hence breaking dormants in buds.
All right, let us also give others. Okay, Ring, please unmute yourself and speak.
They stimulate rapid cell division and elongation in dwarf plants.
That is very, very right. So I don't want to take too much time in this question. So basically, the content that we are trying to talk about here is that we have several plant hormones, and they form various functions. This could also be an assay that we need to be very, very careful about. So for gibberellins or gibberellic acid, it does the following. One is that it promotes cell division and elongation in dwarf varieties, just like Ring has said. We have parthenocapy also, just the same as auxins. Formation of side branches, that is lateral branches, or it ends dormancy, what is promoted by auxins, and also inhibits development of adventitious roots, as well as many more other functions that we might have. So let us proceed to question number 4. Unless someone has a question. If you have a question, please always try and raise your hand up. So question number 4 reads, I see Saint has a question. Yes, please, Saint. We can hear you. Can you speak, please, very fast so that we move to the next number?
So about the functions of auxins, you said that it causes apical dominancy, so that to do what?
So I was trying to say that because it causes apical dominance.
Yeah.
And what do we mean when we talk about apical dominance? Auxins are always highly saturated at the shoot tip and also, of course, at the root tip, the growing meristemic tissues that we have. So at the shoot tip, it promotes just basically primary growth, where we will find that the shoot grows up straight, but there is no development of lateral branches or side branches. In that order or in that manner, a plant will just grow straight without having or ever having branches. And that is because we have high concentrations of auxins in the shoot. So if you decapitate that shoot, if you remove that shoot, just like in coffee or tea, or even in some vegetable varieties like cowpeas, if the shoot is decapitated, then there is formation of the lateral or side branches. I hope you are answered.
Yeah, thank you.
You're most welcome. I can see other hands up. Yes, Evans, you can unmute very fast.
What if I am asked how auxins are used or are utilized as selective weed killers in agriculture?
What if you are asked if
How are auxins utilized as selective weed killers?
How are auxins utilized? You are supposed to explain, or what do you really mean?
You're told explain how auxins are utilized as selective weed killers in agriculture.
So, well, we have types of weed killers in agriculture. We have non-selective and selective weed killers. So if it is a selective weed killer, yes, that simply means that auxins in their formulations in those pesticides, or sorry, not pesticides, but herbicides, in their formulations, they are only used to target specific plants, whereby the auxin part of that plant is going to be, or that formulation is going to target a specific system in that in selective plants in that farm to interfere with distribution of auxins, and therefore it will affect the growth of that plant in general. That is basically how selective.
Okay, thank you.
Yes. Boniface, you have a question.
Excuse me.
Yes, please.
What are the adventitious roots that we talk about?
The adventitious roots.
Yes.
So I'll ask you first, where do we always develop lateral roots from?
The main root.
In a cross section of the root part. We have various parts like the epidermis, the cortex, the endodermis, the pericycle, and others. From those parts that I've just listed, where do we have the lateral roots developing from?
The outer layer.
From those that I've mentioned to you. From all those that I've mentioned, like the pericycle, the piliferous layer, the cortex, and all those. Where do we always develop lateral roots from? So lateral roots, we develop them from the pericycle, that is just close to the xylem. And when we talk about adventitious roots, I do believe, Boniface, that you have seen a maize plant.
Yes.
In maize plant, that is a very, very good example. In maize plant, we always have those roots that grow above the ground surface that resemble, they are roots that grow above the ground surface, and they resemble the roots that we have. Those are the ones that we call as adventitious roots.
Yes. Thank you.
Those ones are the ones that we call as adventitious roots.
Thank you.
Let's now proceed to number 3. I know others may have some questions, but let's also at least cover something as we speak. So question number 4. Science club members designed an experiment as shown below. Examine it. The setup was kept at room temperature for 1 week. And we have those questions. I can still see some people are having more questions to ask. Maybe you can put them in our side chat. Then if we take a break after this, then I can be able to answer them as we proceed, because that means we will be taking too much time in answering same kind of questions. You can put them in our chat, and of course, I'll be able to see what you are putting there on the chat. Thank you. All right, question number 4. The Science Club members designed an experiment as shown below. Examine it. So we have our setup here. Of course, setup A and setup B. What was the aim of this experiment? Any responses? Any responses? You can raise up your hand. Yes, I can see so many people. Yes, Okiring?
To show that oxygen is necessary for germination.
Yes, to show that oxygen is necessary for germination. Why would you say so? Yes, someone else. We have other participants here also raising up their hand. Yes, 26 to 69. He doesn't want to unmute. I don't know what the problem is, because I've asked him to unmute, but he can't unmute. Yes, we have other participants here. We have, yes, we have Leon Lewis. Leon, you can unmute. Leon is not responding. Yes, we have Victor. Victor, yes.
Pyrogallic acid absorbs oxygen present in the experiment.
That is very, very right. That is why we are testing if oxygen is required in this process of germination. Part B, what observation would be made after 1 week? What observation would be made after a week? Not the same hands every time. Yes, at 26, at 26,772. Is not speaking. I don't know why. Yes, Rambui.
In B, germination occurred, while in A, germination did not occur.
That is very good. Can you explain your observation? Why are you saying that is so? Rambui, before you go, why do you say that is so?
Because in A, pyrogallic acid absorbed all the oxygen, so they did not germinate, while in B, all factors were available, so they germinated.
Okay, that is very, very good. For A, they were deprived of oxygen, and in B, oxygen, it's not taken from the experiment. Question number C, why were the seed socks used, why were socked seeds, used in the experiment instead of dry seeds? And that is 2 marks, so you need to give a better explanation for you to score. Leslie? He has lowered his hand. Yes, we have Maina. These guys are lowering their hands when I want to ask them to speak. Yes, Skylar White. You can unmute and speak.
Soak seeds were used instead of dry seeds because soaking starts the germination processes as the seeds absorb water, which activates enzyme for respiration and growth.
That is a very good explanation. So we know that there are so many functions of water in the process of germination. That is just but one of them. There are others also that we might think of, and also those can easily score in that type of response. Part D, name 2 factors that would affect availability of the factor being investigated. What are the factors in the environment that would affect availability of oxygen? Yes, now I can see Leslie's hand is up. He keeps on lowering and raising. Leslie, yes, speak.
Temperature.
How? Explain how temperature influences availability of oxygen. He can't be able to explain. Denzel?
Can you say respiration is a factor here?
How does it influence availability of oxygen? Not really. Thank you for the good trial. Yes, I can see I am stupid. Eh? What is this? What is this? Is a very weird type of name. Now he has lowered his hand. This is a game, and I don't like this game you are now playing. I don't like it at all. So one of the factors that is affecting the availability of oxygen is atmospheric pressure, and also we talk about altitude. An increase in altitude will always affect availability of oxygen. Yes, 269.
Atmospheric pressure.
Yes, we have atmospheric pressure and also altitude, and that is answered. So I'd want to ask you to write the following. When we are dealing with experiments, please let us note the following. Number 1, the aim of of conducting an experiment. So most experiments, you can write and scribble something down. Most experiments, they test on processes. So in a process like transpiration, in a process like germination, in a process like photosynthesis, transpiration, and others, these processes are affected by various factors. So number one, we talk about the aim of an experiment, whereby we use these words to investigate, to determine, or maybe in food test, we always talk about to test for something. So we use that word to. And these things that we are testing for, majorly, they are processes. And I also want to mention that these processes are influenced by various factors. So sometimes, instead of talking about maybe the rate of transpiration, to investigate the rate of transpiration, we can be talking about how maybe to investigate how or to investigate or determine how maybe wind influences the rate of transpiration as a factor. Then apart from aim, please let us go to number 2. We can talk about the various procedures. Sometimes in a procedure you are asked, why would someone maybe in an experiment to test for starch in a leaf, why would you boil the leaf in methylated spirit? So those steps, at least we need to know theoretically when it comes to paper 2, how we deal with those various procedures. Number 3, the reagents that we have in which we use in experiments. Something like calcium hydroxide. If you see that reagent there, already it will give carbon dioxide away. It is used to test for carbon dioxide. Something like sodium hydrogen carbonate is used to donate carbon dioxide in experiments. And those reagents, many of them, they have reasons. In food test, why would I use HCl or dilute HCl in testing for non-reducing sugars? Because it hydrolyzes food, they have various importances. Second, lastly, control for experiments. How would you set up a control for an experiment? And then lastly, once we know the steps or those procedures and why we conduct them, we can always make observations and give scientific explanations. That is in accounting. How do we make observations? 1, observations can either be made by feeling something, like you touch and feel. If it is a cube placed in a hypotonic solution, of course it will be hard. If it is placed in an hypertonic solution, it would be soft. You can also observe by seeing how. You can see bubbles, you can see color change, or you can even see a white precipitate. So we have so many arrays of making observations. You can make an observation by seeing that the level of maybe a liquid in a container has dropped. And then how do we now give the scientific explanation in accounting? Once we know the reasons why we conduct the various procedures that we do. In that procedure, maybe when you add HCl to a non-reducing sugar, we will say that hydrochloric acid hydrolyzes starch to maltose, and therefore maltose is the one responsible maybe for this color change, etc., etc. So those are the main things that we need to check while doing experimental questions in biology. Question number 5. Give 2 advantages of metamorphosis in insects. Yes, I can see someone. Karoki B, yes, you can unmute.
Metamorphosis is used to reduce competition since different stages utilize different types of food.
That is very good. Yes, another response from Miguel. Miguel, you can unmute.
Allowing time for development to occur.
Sorry?
allowing time for development to occur.
So it allows time for development to occur. There are so many other reasons why metamorphosis is important. Why are others not speaking? Why are others not speaking? So when we talk about metamorphosis, yes, I can see someone here. Okay, let me just answer this. When we talk about metamorphosis, it's like you have maize in the farm. And this maize in the farm, when you harvest it, you can harvest it raw and boil it, or you can wait for it to dry and, of course, make some flour. So when we talk about maize flour and boiled maize or roasted maize, those are various forms, but of the same thing, that is maize. So when we talk about metamorphosis, we have various forms in a life cycle of an insect. And we have two types of metamorphosis. Of course, we have complete and incomplete. So we have various life forms. So you can always try and look at those various life forms, the ones that we have, and see how maybe a particular stage is important in the groups, sorry, in the life cycle of that organism. So one, it helps to reduce competition, as in, for example, like our friend has said, it reduces competition in a manner that the larvae and the adults, they eat different types of food. Also, metamorphosis allows time for development and also enables, like pupa stage, enables those organisms to escape unfavorable conditions. Part B, name the hormone responsible for metamorphosis in insects. Name the hormone responsible for metamorphosis in insects. Here, I really would like to see your responses. Yes, 386. You can unmute. 386, you can unmute.
Juvenile hormone.
Juvenile hormone, that is a good trial, though not it. Yes, we have someone, Evans Adino.
Ecdison.
What did you just say?
Ecdison hormone.
Okay, Ecdison hormone. So please, I know some people would want to talk about molting hormone. Look at the question. The question is asking, name. When we are told to name, we don't give descriptive terminologies. We talk about the name, that specific name. Some instances where we always get it wrong, for example, when we are given a guard cell and we have the stoma there, or the stomata being the part that is supposed to be identified, most people, when they are asked to name, they always talk about stomatopore, which is totally descriptive and not a name. There is this one that also I would like to bring to your attention. The hemolytic disease of the newborn, that is erythroblastosis fetalis, where when we have a rhesus negative mother who by chance gets a rhesus positive or bears a rhesus positive child, they always will have the first pregnancy being successful, but the subsequent ones, if there is no medical intervention, there will always be issues. When we are told to name such a disease, we don't go for hemolytic disease of the newborn. That is descriptive. But rather, we go for erythroblastosis fetalis. So the hormone here responsible for metamorphosis in insect is called ecdison. The other hormone, the juvenile hormone that our friend said first, that is responsible for formation of the larval cuticle. And that larval cuticle inhibits molting. It prevents molting to occur past the larval stage. So that is why we disqualify juvenile hormone and we go for ecdison hormone. Question number 6. Distinguish between hypogel and epigeal type of germination. Distinguish between hypogel and epigeal type of germination. Yes, I can see Andrew here. Andrew, yes? You can unmute.
Epigeal germination is where the cotyledon during germination it moves out of the soil, while in hypogel germination the cotyledon remains below the ground.
That is good, and but I would really appreciate a more concrete scientific explanation. Where are the hands? That is very good. Of course, what you've said is true, but we can have other additional information from that. Anyone else? Yes, I can see OT here. Yes, OT. You can unmute yourself and speak.
So you can see in epigeal germination, so we can say
I'm
in
listening.
epigeal germination,
Yeah,
the
listening.
cotyledons are lifted. Or both the cotyledons in epigeal germination are lifted above the ground, and the hypocotyl elongates, and it occurs in dicot seeds.
That is very
While
good,
in
OT. But
hypogel,
that is very good. But before you continue, OT, maybe allow me interrupt a little. Don't mute yourself. When we talk about this type of germination, in the order of events, how you are explaining it, I do believe that the part first elongates, and that is what brings about movement of the cotyledon above the ground. So in your own words, the way you've said it, can you please redirect that so that at least we have a good flow of events? The part elongates first to bring about a change. So please start as you distinguish them.
in epigeal germination, the hypocotyl elongates, hence the cotyledon is lifted above the ground.
That is good. What about epigeal?
In hypogel germination, the cotyledon remains below the ground.
Yes, and which part will elongate? I think you are not looking at the difference clearly. Remember, we are told to distinguish, and if you don't bring out the difference clearly, everything else is going to have an issue. I can see Peter. Yes, Peter?
I had a question on the part where we're distinguishing.
Yes, please.
Is it correct if we distinguish, we give both characteristics of hypogel and epigeal?
Okay. Now, in distinguishing, like our first respondent told us, we talk about epigeal, whereby cotyledon comes above the ground, while hypogel, cotyledon remains below the ground. That is very okay. But at least as a man to have a complete response towards this question, it would be very nice that we talk about, again, the scientific aspect that is involved so that the cotyledon comes above the ground or remains below the ground. For instance, I would say, to answer our question, in epigeal type of germination, we have the epicotyle elongating, pushing only the plumule above the ground surface, while cotyledon, that is hypogel. So in hypogel type of germination, it is the epicotyle that elongates, pushing the plumule above the ground surface, while cotyledon remains below the ground surface. And then in epigeal type of germination, we can talk about the hypocotyl elongating to push the plumule and the cotyledon above the ground surface. And that would give us an undisputed response as Mangu men. So please let us just note in the question number 6 that the difference must come out clearly for you to score the 2 marks. So if you don't distinguish, then you don't score any mark. Question number 7, I hope you answered. Question number 7, state 2 roles of water during the process of germination. I see Hugo has raised up his hand. Yes, Hugo?
Softens the seed coats and facilitates cell elongation and cell division.
Softens the seed coat, that is very okay. Facilitating cell division, not really. Someone else, DNG?
It facilitates the hydrolysis of stored food substances.
It hydrolyzes food. Any other? I see Sami Fulmo.
Water activates the enzymes and provides the medium for the enzymes to act and break down the stored food into soluble food.
Activation of enzymes, provision of a medium where reaction takes place, those are 2 different points, and that is very good. Yes, I can see Ramboi.
It's a medium of transport for dissolved foods.
I do believe that we have said. Now that is like a repetitive question, and maybe we can proceed. Question number 8. The diagram below shows the life cycle of a butterfly. Name the type of life cycle shown above. What type of life cycle is this? And please give us a reason for your answer as you answer that. Yes, Denzel? Denzel, you can unmute.
This one is complete metamorphosis.
Why?
We can see by the presence of 4 stages, which is the egg, the larva, the pupa, and then the butterfly. Yeah.
That is very good. And also you can talk about, there is the pupal stage, because in complete metamorphosis, we don't have the pupal stage. We have egg, nymph, and adult. Alan, you have something to say? Okay, that is answered. As you can see, he has lowered his hand. Okiring has something to say. Part B.
That several eggs are laid.
Several eggs are laid. Is that a distinguishing feature here? Can you see several eggs here? It's not observable, so you cannot see it from our diagram. Yes, BK Kariki, please speak.
The larva is completely different from the adult.
So we have larvae being completely different from adult. And so you'd compare a nymph and also an adult. Well, we can always try and remain positive and talk about things that we easily see. So for instance, you can talk about presence of pupal stage, which is not in complete metamorphosis, and also larvae. And then also we can talk about the 4 stages. And I think those are basically it. Suggest the importance of such type of life cycle. Suggest the importance of such type of a life cycle. Yes, Abdi. Yes, Abdi, speak. Abdi, you can unmute and speak.
This helps. Absence of larval and pupal stages shortens the life cycle of an organism.
Absence of larva and pupal. No, we are talking about complete. This is complete metamorphosis. So how is this advantageous? What you are giving is for incomplete?
It helps to allow time for development to take place.
Allows time for development. Please let us also avoid these words like helping, acting. Those are not biology, brothers. Let's talk about allows time for growth. We can talk about reduces competition, and those are the importances of having the metamorphosis that we talked about. These are various life forms. So using letter symbols list the various stages in the sequence in which they occur. So that is a question giving you direction, and you're not supposed to use names. So here, if you are using symbols, you'll start with letter symbols. You'll start with this is stage J, then we go to stage B, then we go to stage V, then we go to stage M. And that would be that life cycle. So you start with the larvae, then we go to you start with the egg, then larvae, pupa, and all the way to adult. And that is how you give that life cycle using letters. So you use those letters that have been used in our illustration. So of the stages labeled, name the resting stage and the feeding stage. Of these stages that have been labeled, name the resting stage and the feeding stage. Yes, DNG, you can unmute.
The resting stage is stage V, while the feeding stage is B.
That is very okay, and we proceed. What is the importance of stage B and stage V? What is the importance of stage B and stage V? What is the importance of having larvae? And what is the importance of having pupa? The same hands up. What are others doing? They just joined the class and they don't want to speak. Yes, Tim Collins, where did you go to? Please speak and mute yourself.
The stage B, it allows the larva feeds a lot. The stage V, it allows the reorganization. V.
So you okay, that is good. Let us also get responses from others. We have Caro. Please, Caro, you can unmute. Caro, you can unmute yourself and speak. Caro, you can unmute and speak. Seems like he's having issues. Let us go for DNG. DNG, can you unmute and speak?
So stage B allows the organism to feed and store the energy for metamorphosis, while stage V allows the organism to conduct metamorphosis.
That is good. What about what does Denzel have to say?
We can say that stage B, it's the feeding stage because the larva feeds to acquire the energy, like enough food and nutrients for the pupal stage. And in the stage V, the pupal allows for the reorganization of tissues.
That is very good. So for stage B, we can talk about feeding to store or to have sufficient energy that will be required during cell reorganization in stage V. And also because this stage has locomotory structures, just like we have the adult stage M. These two stages also allows for dispersal because they can move from one place to another. While stage V, like just we have said, this is that dormant stage. And in this dormant stage, it allows for development. And when you talk about development, you know that is an irreversible change in complexity. So it allows for development to occur. And after that development occurs, of course, there will be an adult emerging from that stage. So those are the right responses that we have. Question number 9. The graph below represents the growth of animals in certain phylum. Name the type of growth pattern shown on the graph. Identify the process X and name the hormone responsible for the process in B above. So again, we have responses. Where others? Not the same hands every time. What are others doing? Yes, I can see someone called Free Palestine. He has again lowered his hand. I need new people to also speak. Not every day, every time we have the same hands up. Yes, we have Didier Deschamps. Please unmute yourself and speak.
Process W is the growth phase.
We have not been asked about W. Name the type of growth pattern shown on the graph. He has gone MIA. Where are others? Yes, I can see Hakim. Hakim, please unmute yourself and speak. He's not able to unmute himself. We have AKK. Unmute yourself and speak. I can see someone doing some mujanja here. Is calling himself intermittent curve. Intermittent curve, please unmute yourself and speak. He's doing some mujanja here and there. So these people, they don't want to speak or what? Well, this is an intermittent curve, and that process X is where molting occurs. And of course, that hormone is called ecdysone hormone. Question number 10. The diagram below shows some growth in a plant. Name the type of growth shown in the diagram. Responses. Let me see our chart if there are any responses. There are no responses in the chat. I can see people. Yes, Larry?
Secondary growth.
This is secondary growth. That is very good. Name the part labeled A. Yes, Njogona?
Secondary phloem.
That is secondary phloem. Really? Not really, not really. Good trial though. Yes, I can see Leslie. Leslie, you can unmute yourself and speak. He doesn't want to speak. Yes, Andre? All right, please
primary
let's
phloem.
that is the primary phloem. So
Primary phloem.
let us note this as we discuss about this cross section. Let us see and let us always note that this is our cambium ring, this cycle here, and towards the inside, of course, we will be having the xylem. I hope you can see my cursor. And of course, towards the outside, we are going to have our phloem of this cambium ring, this part B. So when growth occurs, that is when secondary growth occurs, to the inside, we are going to form new cells that is going to push the old cells deeper inside towards the pith. And then to the outside, we are again going to have new cells forming just adjacent to this ring. Therefore, we are going to have the old cells being pushed further away. So what do I mean the old and the new cells? The old are the primaries, while the news are the secondaries, such that the primary xylem will be deeper inside towards the pith, while primary phloem will be to the outside next to the secondary cortex. So their secondaries will be just around this cambium ring where my cursor is pointing. So what is the function of part labeled B? Functions of part labeled B. Functions of part labeled B. So I can see hands up. So we have a new face here. That is Jimmy. Jimmy, your response? You can unmute yourself and speak. No, he has gone MIA. I think he has issues with his mic and maybe network. We'd go for someone else. Alan, unmute yourself and speak.
It forms cells for secondary thickening.
So it is the region that mitotically divides to bring about secondary thickening. That is good. Question number 11. The graph below shows a sketch of growth pattern of the femur of an animal. So we have length of femur and time in days. Identify the growth curve shown in this diagram above. Mwangi?
Intermittent.
That is intermittent growth curve. Name the phylum to which the animal whose growth pattern is represented in the graph. Which phylum would be this? We have two quadricics.
Arthropoda.
That is phylum Arthropoda. I know someone would talk about phylum Chordata because he sees femur here, especially for those who are for Form 4s. They always would want to do that because they see femur and they may be thinking that femur is only found in human beings. Of course, even insects, they have femur. All right, so that is phylum Arthropoda. Account for the rate of growth of animal during the period labeled J. So we have that period labeled J. Account for that growth. Yes, responses? Now, when it comes to accounting, no one is willing to speak. I can't see any hands up. People are now so quiet. Why are people so quiet? Account for the rate of growth of the animal during period J. Yes, someone wants to speak, DNG?
So during period J, the cells divide, but the length does not increase due to the inhibition by the exoskeleton.
That is a very good one. Any other perspective towards that? Yes, Abdi, you can unmute yourself and speak.
After molting, growth occurs rapidly until the skeleton hardens. The growth rate slows down as the exoskeleton hardens.
Oh, that is talking about both part I and J, so it's not giving out clearly what we really want. So please remember, in accounting, you talk about an observation. An observation is that growth is constant. So you need to give us a scientific explanation why growth is constant at that time. That is what we really want. Because time J, you can see there is no growth at all. That is what we want us to explain. Why is there no growth?
So the rate of growth remains the same in period J because of the presence of the exoskeleton, which prevents further growth.
That is it. And you hit it. You hit it right. So we don't have growth here in part J. And that is what we are told to account for. And that is because we have the exoskeleton limiting growth. And of course, at the end of J, where my cursor is pointing, we have molting occurring before again that exoskeleton hardens. That is in Roman 2, hardens to again stop growth for a while. That is what we are trying to talk about. So we have only been told to account for the rate of growth during period J. What is the observation? Change in X causes no change in Y. So there is no growth occurring. And that is why we're talking about the exoskeleton. So explain the importance of the process named above, the importance of this process. So I've skipped this. Name the process that occurs at point I. Name the process that occurs at point I. That is Roman 1. I can't see answer. Yes, I can see Didi Saho. You can unmute and speak.
The process is growth.
The process is growth. I give a reason.
This is due to increase in the size.
Of course, the process that we are testing here is growth, right?
Yeah.
But does that D1 now represent growth? Because growth is represented by this curve, right?
Yeah.
So is that process 1 really growth? So that is a good trial. Someone else, I can see Emmanuel. Oh, these students.
Process is molting.
So the process is molting or ecdysis, and it is caused by molting hormone. I can see other people here raising up their hands and they have funny names. I won't read those names out. So you can go with those names and maybe play with them somewhere, not in this serious class. So that is ecdysis or molting, and that is done by ecdysone hormone. So we move to question E. Name the type of growth in the animal where some parts of the body grow at a faster rate than others. Name the type of growth in animal where some parts of the body grows faster than others. What would be that type of growth? Any ideas? Yes, I can see John raised up his hand. John, you can unmute and speak.
Secondary growth.
Secondary growth, that is a good trial. Not really, it's a good trial. Any other? I can see Moses has raised up his hand. Yes, Moses?
Can we still say intermittent growth?
Not really, though a good trial. I see this one is challenging us. Any other trial? Yes, I can see Victor. Victor. Yes, Victor.
Yes. Metamorphosis.
Is metamorphosis a type of growth? Not really. Thank you for that. Any other last trial? The very, very last trial.
Discontinuous growth.
Yes. Sorry?
Discontinuous growth.
Yes, that is a discontinuous type of growth, and that would be the response in that. Question number 12, state the functions of the following parts in a germinating seed. We have coleorizer and coleoptile. Yes, I can see a new face here. Isaac, unmute and speak. He doesn't want to speak. Maybe we have Techno BG7. No, these guys, they don't want to speak. I don't know why. Yes, you can speak Techno BG7, yes.
Or protect the radicle from mechanical damage.
That is which, coleorizer or coleoptile?
Coleorizer.
That is good. What about coleoptile?
Protect the plumule from mechanical damage.
That is very right. The photographs below show 3 bean seedlings that are of the same age, but were grown in different environmental conditions. Based on the external appearance of the seedlings, suggests the condition under which these were grown. So this is a concept in Form 4, and maybe if someone has read ahead, maybe they can try, but it's a concept ahead. Because these questions are for all growth, you can also invite concepts from other parts. So I can see Isaac still has his hand up. Isaac? He has lowered his hand and disappeared. Yes, I can see number 12. Please speak. You can unmute and speak. He's not speaking. We have Aldi TKN. Yes, Aldi?
Okay, so I can say seed Q was grown under one-sided light.
Proceed, that is good.
Seed R was grown under adequate light.
That
while
is good.
Seed S was grown in the dark.
That is also very, very right. You have nailed all of them, and this is a concept we always get in response coordination and exception in Form 4. That is good. That is a very good one. So for seedling Q, that was grown where light only was coming from one source, or what we can call as a unidirectional light source. So it will grow towards light, while R was growing in a place where light is evenly distributed, while S, that phenomenon, we call it as etiolation, because it is growing long and slender so that it is able to reach where light is. So when we told under what conditions, we talk about light from unidirectional source, equal distribution of light or even distribution of light, and then we can talk about in a dark area. Give 2 differences between R and S. Give 2 differences between R and S. So I can still see okay, can see new hands, and this is good. I can see name a junior. You can unmute and speak. Neymar, you can unmute and speak. Neymar is not speaking. Let's go for OG. Yes, OG, you can unmute and speak.
R is shorter, while S is tall.
R is short while R is tall. We have Ryan Moti.
We can talk about the leaf size. Leaf R has a dark green leaves, while leaf S has small and pale leaves.
That is very good, although we can't see the color here, but that is a very good theoretical concept kind of knowledge. That is very good, but it's very, very theoretical because you can't see the colors here in the diagram. I hope that is right.
Yes.
But it is right, but theoretical. Yes, I can see other people having funny names. I won't be calling out those names. So you either change them or I won't be naming them. So we have Jimmy. Jimmy, you can unmute.
R has lateral buds, and S does not.
Really, this is just a small seedling. That is a good child, though. Let us also hear from others. I can see Andy Mwangi. Andy Mwangi, I believe Andy is from the 3 Duke. Yes, Andy?
Can you say something to do with photosynthesis?
You please lay out your ideas so that at least we also get them.
Okay, like I was saying, R, you can say photosynthesis occurred, but S did not.
Okay, that is a good one, and that is a very good trial. This person called Diogo Jota. I know Diogo Jota is a footballer who passed on some months ago. So I won't be mentioning such names, and I won't unmute you. So, well, this is a theoretical concept, and which can also be practically applied, and it is in response, like I mentioned. So the differences between these two plants include one, this one has a thicker stem while this one has a slender stem. The other difference could be in the leaf sizes. Of course, this one has broader leaves, these ones will be having smaller leaves. And also, lastly, we can be talking about their color, which someone has mentioned. The guy from Dayton. We can talk about this one being green in color, while this one having pale colors. And that goes for both the stem and the leaves. Then question number 14. I see we only have 9 more minutes to go. We do question number 14, part A and B. And if there are any other questions that we might want to ask, we can always, we can raise our hands and then ask, and then we call it a day for today. So question number 11A. Identify the type of germination exhibited by seedlings A and B. I can see Isaac D, yes? Isaac D, you can unmute. He has gone MIA. I can see someone by the name A is epigial, B is hypogial, and that is the correct answer, but you can unmute and speak so that also we can hear what you have to say. Yeah, he has also gone MIA, and that is good. Those are the responses. A is epigial, while B is hypogial. So I want to stop there. And in any case someone has questions, you can put them in our chat, or you can equally raise up your hand. So I'll stop sharing my screen so that at least I can answer your questions. So let me see what we have in the chat first. So in our chat, we have, let me see what we have here in our chats. Someone said that they can't see my screen. I was sharing my screen. Maybe that was an issue with your network. Next time, maybe you can have a good internet connection. From the chat side, I can't see questions. Oh, someone called Larry Mburu is asking, what is the difference between allometric and isometric growth? The difference between, someone can be writing this down, isometric and allometric growth. That is a very good question, Larry. So when we talk about these types of growth, isometric and allometric growth, these guys, they describe how various or different parts of an organism grow in relation to other parts of the body of that same organism. So when we talk about this as isometric growth, it is whereby all parts grow and increase at the same time. That is, they grow proportionally. While when you talk about allometric growth, that is where different parts of the body of an organism, they grow but at different rates. That is what we have as the difference between allometric and isometric growth. So I want to quit the chat because that is the question that I've seen, and see people who have raised up their hands so that maybe they can ask questions. If you have any other question, you can raise up your hand. Where is the juvenile hormone produced? That is a question from, I believe that is DNG. Juvenile hormone is produced from the corpora alata. So corpora alata, those are the pairs of glands that we find in the brain of insect. Kama ni moja, we call it as corpora alatam. Kama ni mingi, we call them as corpora alata. And also that reminds me of something else. Someone might want to ask you, why is it that adult insects, they don't molt past that adult stage? Mbona zisichange past hiyo adult stage zikuwe kitu ingine? That is because, and if someone is noting it down, the prothoracic glands that secretes the molting hormone, they disintegrate at some point so that the adult cannot molt past that stage. I can see someone raising up their hand here. This is Aldi. Aldi, you can unmute and speak or ask your question.
Okay, so the question which you asked about the seedlings being grown under different conditions.
Yes, please.
The question about differentiation of the seedling R and S. So can you talk about how do you explain the difference in relation to the release of auxins?
Okay, so that again is in response, that is in form 4. But to answer you so that you don't be left hanging, allow me just share my screen again, and then I'll use that diagram to explain what is it that I'm giving out. I do believe you can see my screen.
Yes.
Good. So that diagram is here. So in response to your question, we have Q, R, and S. Then Q is growing towards light. And how do we explain that scientifically? So number one, we need to agree that auxins are always found at the shoot tip and the root tip because they promote primary growth, cell division, and cell elongation.
Yeah.
That is one. Secondly, auxins are always sensitive to light. So if light is coming from one direction, auxins will diffuse to this other side that is not receiving light of that same stem. So kama light inatoka east side, then they'll diffuse towards this other region which is not receiving sunlight, because we are talking about light diffusing from one direction. Therefore, this other side which is darker, we will be having more auxin concentration. Therefore, more rapid cell division and cell growth in the darker side than the side which is having more light. Therefore, this darker side grows faster than the side which is having more light. Hence, the curvature or the plant curve or bends towards light. While in R, because the light is coming from all other parts, in R, there will be even distribution of auxins, and that seedling grows straight up. And I believe that is the response to the question that you've just asked. Are you answered?
Yes.
Good. The last question I'm taking, because you have only 1 minute left, is from 26051. Please ask your question. You can unmute and ask, please.
For clarification. So
yes,
what was the name of the chemical substance used in plants that helps in flowering of plants?
Helps in
flowering in plants.
So the plant hormone, I do want to believe that the plant hormone that facilitates flowering is florigen. Yes, the last question from yes, sorry. The one that
helps in leaf fall.
Leaf fall or leaf abscission.
Sorry, leaf
that is abscisic acid.
Okay, thank you.
All right, Evans Adino, last question. Time is up.
What was the? May you pardon the importance of the larval stage?
The importance of larval stage. The importance of larval stage is that larvae always feeds a lot, and that feeding a lot, because the pupa does not feed because it's always in that puparium. It gets a lot of food that is going to be used during the development or rearrangement or reorganization of cells.
And which is the resting phase? Resting phase, sir.
Yes, that is the resting
which is
phase.
Yes.
Which is the
feeding stage and the resting stage.
The feeding stage, like I was saying, is larval stage.
Yes.
The feeding stage is the larval stage. While you can listen as I speak, so that when I'm done, maybe you can respond. So the feeding stage is the larval stage, while the resting stage is the pupal stage. And
I'm requesting if
yes.
I'm requesting if you could send to us this paper that we have gone through, please, to the parent group.
All right, I'll liaise with your teacher so that we can see how that happens. Thank you for being good gentlemen, and thank you for having that interactive session. Have a good day ahead. Bye-bye.